So you're going to be learning some naming conventions. Big one is, how do you know? How do you orient yourself to figure out which strand is which? Because that's the core. If you don't have that, you're always going to be confused. And then which terms are interchangeable? And which aren't? And the big thing, which is what this entire video, well, the majority of this video is about, is what does antisense mean? What does sense mean? What is the coding strand? And what is the template strand? Some other important things to know and get a handle on as we're moving in to talking about how we produce our gene products is how the strands of DNA are named, because this is sort of, there's some weird, confusing terminology if we don't sort that out. One of the first things to know is that just by convention, it's just the convention that developed that gets used. The the top strand is drawn as 5' to 3'. We know this is not necessarily how DNA is oriented, right? We talked about that it's chromatin. We talked about that it looks like, we envisioned it as spaghetti. This is just by convention. This is how it's drawn. So the complementary strand, this strand that it's going to be hydrogen bonded to via its base pairs, what are these going to be? Stop and think for a second. What will this end be, and what will this end be? Okay, hopefully that popped right in your head. The complementary strand is going to be, if the top strand is 5' to 3', the complementary strand is going to be 3' to 5'. Also recall that genes are encoded by both strands, and there's some terminology that is specific to the gene of interest. So meaning that the naming of the strands will change if the gene of interest is on the other strand. I'm going to note that. So the naming of the strands will change if the gene of interest is on the other strand. And hopefully that will make a little bit more sense here in just a second. So let's imagine that our gene of interest here is this area that I've colored in pink. Our gene of interest is on the 5' to 3' strand. Cool. The strand with the gene of interest is called the antisense strand. It is the antisense strand. So this gene that we're interested in is on this 5' to 3' strand. Therefore, this strand is currently called the antisense strand. The strand, the complementary strand, is the sense strand. I'm going to make the joke. I just can't help myself. That doesn't make any sense. Kind of seems like the strand that has the gene of interest should be the sense strand, not the anti-anything strand. So let's pick that apart and make it make sense. Explain why. So let's imagine that our gene of interest has this particular code. Okay? Adenine, thymine, cytosine, guanine, guanine, thymine, cytosine, guanine, cytosine, thymine. This is part of that gene. And let's say we want to know what the mRNA is for that. What's the mRNA code going to be? So you may recall that what I told you to do was figure out what the complementary DNA strand is going to be, because the mRNA is almost exactly that complementary strand. This is why we call the complementary strand to our gene of interest the sense strand, because we can just read the sense strand and immediately know what the mRNA is. So sort of like I taught you to not just go directly from the gene that we're trying to encode to mRNA, but first go through the complementary strand. This is why it's called the sense strand. This makes even more sense when we remember, and we're going to go into this in more detail, that the mRNA itself is what actually encodes the amino acids that make the proteins, right? So the mRNA encodes the amino acids that make up a protein. So the mRNA is what can tell us what's going to be built. In the cases of genes that aren't going to become a protein, that are just RNA, then that's the actual code we're interested in, right? So the sense strand allows us to determine what the codons are. Or in the case where it's a gene that isn't turned into a protein, isn't translated into a protein, then it just tells us what the product is directly. And so that's why this is called the sense strand. And the strand that actually is the code, is the template for the mRNA, is called the antisense strand. Okay, so more terminology. The antisense strand is also called the template strand. It's called the template strand because it is the template for mRNA, right? It's the template for mRNA. But mRNA is complementary to that strand. So the sense strand then is called the coding strand. It's called the coding strand because it tells you what the mRNA code will be, or the mRNA or RNA, if it's going to be an RNA gene, code will be. So this means that you can just read the sense strand or just read the coding strand. Those are the same thing. And that immediately tells you what your codons are going to be, or it tells you what the code of your RNA gene is. It tells you what the product is. So that's why these have these sort of weird, seemingly at first glance, backwards naming. Okay, so now let's think about our DNA. And let's imagine that we have this section of DNA here that has our gene of interest, and we want to transcribe it. And don't worry, we're going to get into all the details of that, how that happens. But this sort of introductory core information, I believe, will help you not get confused as we work through that. So here's our gene of interest, and this is our 5' to 3' strand. And then the complementary strand, of course, is 3' to 5'. So our mRNA that gets made from this DNA, this template strand, remember this is our template strand, also called our antisense strand, is the one that encodes our gene. When we make mRNA, I think I used white before. Okay, our mRNA is going to be complementary, right? So which direction does it run in? Stop and think about that for a second. What are its primes going to be? So what would you put at this end, and what would you put at this end? It's complementary. So this end of the mRNA is going to be 5 prime, and this end of the mRNA is going to be 3 prime. And I'll label that. This is our mRNA. Okay, so these are just some important things to have in your head as we start digging into the way that transcription